Welcome back to the deep dive. Today we are looking at a virus that everyone thinks they know.
Yeah, everyone thinks they know it right up until they actually get the real deal.
Right, exactly. Because you know the scenario. It is Tuesday. You wake up at 7 in the morning, you have your coffee, and you feel totally fine.
Maybe a little tickle in the throat.
Yeah, maybe a tickle, but you push through it. You go to work. But by 11, you feel a little off. And by 1 o'clock, you are shivering at your desk.
And by 3, you feel like you have been physically run over by a semi-truck.
Hit by a truck. That is always the phrase. It is not just a bad cold. We are talking about the Orthomyxovirus family today, the true influenza.
That hit by a truck feeling, that is the classic textbook presentation. We call it prostration.
Prostration, like you literally cannot get up.
Exactly. It is not just I feel gross. It is I cannot move my body from this bed. High fever, severe myalgia, so deep muscle pain, and a headache that just feels like your skull is in a vice.
So we are definitely not talking about the rhinovirus sniffles today. We are decoding Murray's Medical Microbiology, Chapter 49.
With some heavy tactical support from Jorwitz, Chapter 39 as well.
Right. And looking at this stack of research for this deep dive, it honestly feels less like a biology lesson and more like we are analyzing a military campaign.
It is absolutely a war. And the reason this specific virus is so fascinating, and frankly why it is such a massive staple of medical board exams, is that its physical structure dictates its global strategy.
Structure dictates strategy.
Yeah. If you take one single thing away from this deep dive today, let it be the central concept. Segmentation equals adaptability.
Segmentation equals adaptability. I like that. It sounds like a tech startup slogan, but, deadly.
Deadly and incredibly efficient. The way this virus is built allows it to hack its own evolution in a way most other viruses just simply cannot do.
Okay, before we get into the microscopic mechanics of that, let's set the board for everyone listening. Who are the actual players in the Orthomyxoviridae family?
Well, we have 3 main genera that impact humans. There is influenza A, influenza B, and influenza C.
Just A, B, and C. Simple enough.
There is a 4th one called Thogotovirus, which is a bit of an outlier, but A, B, and C are the ones you will see in the clinic and on your exams.
And the name itself, Orthomyxo. I mean, myxo always sounds like a sticky situation to me.
It is literally sticky. The Greek root muxo refers to mucus.
Oh, of course.
Yeah, these viruses were originally named because virologists noticed their really high affinity for mucoproteins. They bind to sialic acid, which is a key structural component of the mucus lining your entire respiratory tract.
So right from the jump, the name is telling you exactly where it wants to live, in the slime.
In the slime, exactly.
Lovely. So we have A, B, and C. For the students listening right now, how do we categorize them? Like, which one is the world ender and which one is just an annoying Tuesday?
Influenza A is unequivocally the world ender. It is the most virulent, it causes the most severe clinical disease, and crucially, it has a massive animal reservoir.
When you say massive, what are we talking about?
I mean, it infects humans, obviously, but also birds, pigs, horses, seals, whales. It is everywhere.
Whales. Really?
Yeah, aquatic mammals are absolutely susceptible. And because it can jump between all these different species, influenza A is capable of something called antigenic shift.
Yeah, it's genetic shift.
Right. Capital S shift is the molecular mechanism behind massive global pandemics.
Okay, so A is for animals and A is for apocalypse. Got it. What about B?
Influenza B is primarily just a human pathogen. It does not have that vast global animal network to swap its genes with.
So no animal reservoirs means no sudden massive shifts.
Exactly. So B does not cause pandemics. However, you really cannot underestimate it. It causes very significant seasonal epidemics, and the clinical disease can put you in the ICU just as fast as A.
It is just evolutionarily slower.
Yes. It only undergoes antigenic drift. Capital D, minor changes over time.
Okay, so A is the pandemic shifter. B is bad, but basic, it just drifts. And what about C?
Influenza C is the mild cousin. It usually just causes a simple upper respiratory illness very similar to a common cold.
So a lot of people probably get C and never even know they had the flu.
Most people get it in childhood and just assume it was a regular bug. Structurally, it is distinct too. It only has 7 genomic segments, whereas A and B have 8.
And just to close the loop, what was that 4th one you mentioned?
Thogotovirus. That is the curveball. It is an arbovirus, meaning it is actually transmitted by ticks.
Flu transmitted by ticks. That is wild.
It is very rare, but there was a famous case back in 2014, the bourbon virus in Kansas, which unfortunately was lethal. It is just a good reminder that this viral family is weird and wonderful.
But for the exam, stick to A and B.
Absolutely. Focus your energy on A and B.
All right, let's zoom in now. I really want you to help us visualize this thing. If I had super electron microscope vision, and I'm looking directly at an influenza particle. What am I actually seeing? Murray describes it as pleomorphic, which I am guessing is fancy textbook speak for a shapeshifter.
That is pretty much exactly what it means. Pleomorphic just means it does not have one single rigid fixed shape, like, say, a pox virus, which looks like a brick.
Right, pox is a brick.
With flu, it is usually spherical, roughly 80 to 120 nanometers across. But sometimes, especially when you first isolate a fresh strain from a sick patient, it can be filamentous.
Like long and tubular.
Yeah, like a long tube. But regardless of what the outer lipid envelope looks like, the real magic, the danger is on the inside.
And this brings us right back to your core theme from the start.
Yes.
Segmentation.
Yes. Most RNA viruses we talk about have a genome that is like a single continuous scroll of parchment. One long continuous strand of RNA.
Like if you cut the scroll, the whole story is ruined.
Exactly. But influenza is completely different. Its genome is like a loose leaf binder. Or better yet, think of it as a deck of cards.
A deck of cards. Okay, I can picture that.
For influenza A and B, that deck has exactly 8 cards, 8 completely separate individual segments of negative sense single-stranded RNA.
Why is that an evolutionary advantage, though? Why carry around a loose deck of 8 cards instead of just keeping everything on one nice, neat scroll?
Because you can shuffle a deck. Think about it. Imagine I have a deck of cards. Let's call it the human flu deck. And you have a deck called bird flu deck. If we both infect the exact same cell in the exact same host at the exact same time, we basically throw all of our cards into one big pile on the table.
Right inside the host cell nucleus.
Inside the cell. And when the new baby viruses are being assembled to leave the cell, they do not carefully sort the deck spec out. They just blindly grab any 8 cards from the pile.
So a new virus might come out holding the King of Hearts from my human deck, but the Ace of Spades from your bird deck.
Exactly. And if that Ace of Spades happens to be the instruction manual for a bird surface protein that human immune systems have never seen before.
But the rest of the cards are human cards that make the virus really good at spreading from person to person.
Then you have a brand new global pandemic. That exact process is genetic reassortment. And it is only physically possible because of those individual segmented cards.
That is terrifyingly brilliant. It is mixing and matching its own weapons. But let's look at the cards themselves. What are these RNA segments actually wrapped in? Because they're not just floating around naked, right?
Right. The RNA is wrapped in a structural protein called nucleoprotein, or NP for short.
Yeah, NP.
Think of the NP as the plastic spool that the RNA thread is wound around tightly. This is actually a very high-yield fact for diagnostics because the NP is the main ID badge of the virus.
Meaning what exactly?
When a lab runs a test and tells you, yes, this patient has influenza A, it is specifically detecting that type-specific nucleoprotein. It does not mutate very much. It is the one constant in all the viral chaos.
So NP is the internal skeleton, the identifier. But Murray really emphasizes that influenza is a negative sense RNA virus. And I know a lot of people studying this struggle with the concept of sense. Why does it matter if it is negative or positive?
It is all about how the host cell reads instructions. Think of positive sense RNA as a beautifully printed, ready-to-read instruction manual.
Like an IKEA manual that is already in English.
Perfect. The host cell's ribosomes can look at that positive sense RNA and immediately start building viral proteins. But negative sense RNA is like a photographic negative, or text written backward in a mirror.
So the host ribosome looks at it and says, I have no idea what this is. I cannot read this.
Exactly. The cell cannot translate it. So the virus has a massive problem when it enters the cell. It has the blueprints, but no one can read them.
So it has to bring its own translator.
Yes. It carries a viral RNA polymerase complex. This is made of 3 specific proteins called PA, PB1, and PB2, and it packages this entire complex right inside the virus particle attached to the RNA segments.
So it is basically lugging a heavy photocopier around with it everywhere it goes.
It has to, because the very second the virus opens up inside the cell, that polymerase complex has to get to work immediately converting the backward negative strand into a readable positive strand so the host cell can finally understand what to build.
Okay, so we have the RNA, which is the blueprints. We have the NP, which is the spool holding it. And we have the polymerase, which is the photocopier. That is the inner core.
That is the core.
Now let's talk about the outside, the envelope, because this is where the real weaponry lives.
The envelope is actually just a lipid membrane that the virus steals from the host cell when it leaves. But before it leaves, it studs that membrane with its own viral spikes.
And there are 2 glycoproteins here that you absolutely have to know for any exam.
Haemagglutinin and neuraminidase, HA and NA.
The H and the N, the
Yeah.
letters we see on the news every year. H1N1, H5N1. Let's break them down, starting with the haemagglutinin.
Think of haemagglutinin as a grappling hook. It is a trimer, meaning it is 3 protein subunits twisted together like a strong rope.
And its primary job is just grabbing on.
Yes, it is the viral attachment protein. It binds specifically to sialic acid receptors on the surface of your respiratory epithelial cells.
Murray mentions hemagglutination as a laboratory phenomena. Is that the exact same thing happening in the body?
It is the same biological function, just being observed artificially in a test tube. If you take a sample of influenza and mix it into a test tube full of red blood cells, the HA grappling hooks will indiscriminately grab the sialic acid on the red blood cells and tie them all together into a giant clump.
A lattice of trapped red blood cells.
We call that agglutination. It is how virologists used to diagnose and quantify the flu before PCR was invented. But inside the human body, it is simply how the virus docks with the mothership.
So HA is the grappling hook getting you in. Now what about neuraminidase, the N? You called this one the machete earlier.
Yes, neuraminidase is an enzyme. Specifically, it is a sialidase. Its entire job is to chop sialic acid off the surface of cells.
Wait, hold on. This seems completely counterproductive to me.
How so?
Well, HA needs to grab sialic acid to hold on, right? But NA is chopping sialic acid away. Isn't the virus just fighting itself?
It seems like it, but it is actually a very delicate, brilliant balance. You absolutely need the HA grappling hook to get into the cell. But fast forward to the end of the infection cycle.
Okay, the cell is dying and pumping out new viruses.
Millions of new baby virus particles are budding out of the cell membrane, but the surface of that dying host cell is still completely covered in sticky sialic acid receptors.
Ah, I see. So if you did not have the machete.
The new viruses would bud out and immediately get stuck right back onto the dying cell that birthed them, or their grappling hooks would grab onto each other and they would just form a useless sticky ball of viruses.
So they would be permanently trapped in a sticky prison of their own making.
Exactly. So neuraminidase acts as the machete, hacking through all those sialic acid tethers. It cleaves the receptors so the newly built viral fleet can actually launch and float away to infect the next person.
It also helps them chop through the mucus in the throat, right?
Yes, mucus is full of decoy sialic acid receptors. Na chops through that thick slime layer so the virus can physically reach the living cells underneath.
So the HA gets you into the party.
Yeah.
And the NA clears the exit so you can leave and go crash another party down the street.
That is a perfect analogy. And this specific mechanism is exactly why the drug oseltamivir works.
Tamiflu.
Right. Tamiflu is a neuraminidase inhibitor. It physically gums up the blade of the machete.
So if I take Tamiflu, the virus can still replicate inside my cells.
Yes, it replicates just fine. But when the new viruses try to leave, they cannot cut the tether. They just clump up on the surface of the dead cell, and eventually your immune system comes along and cleans them up.
That is fascinating. So that is H and N. But there's one more envelope protein we need to discuss.
Yeah.
This one is specifically only found in influenza A, the M2 protein.
The M2 ion channel. I call this the escape hatch. Or maybe airlock is a good visualization.
Airlock. Why do we need an airlock?
When the virus first docks and gets swallowed by the host cell, it ends up trapped inside a cellular bubble called an endosome.
Like the cell is trying to digest it.
Exactly. The cell starts pumping acid into that endosome bubble to destroy whatever it just ate.
Yeah.
But the virus actually wants this. It needs that acid.
It's uncoated.
Yes. The M2 channel is a viral pump that takes those acidic protons, the H+ ions from the endosome, and channels them directly inside the viral particle.
So it intentionally acidifies its own core.
It does. That sudden drop in pH dissolves the internal matrix shell, the M1 protein. The shell falls apart and releases the 8 RNA segments into the cytoplasm.
And this M2 airlock is where the old drug amantadine used to work.
Amantadine is literally just a molecular cork. It perfectly plugs up the M2 channel. If protons cannot get inside, the virus cannot dissolve its shell. It stays locked inside its own armor forever.
You said M2 is only in influenza A?
Correct. Influenza B does not have an M2 protein. It uses a totally different mechanism to uncoat. So amantadine never ever worked on flu B.
And frankly, it does not even work on flu A anymore.
Sadly, no. Almost all circulating strains of influenza A have mutated their M2 channel shape, so the amantadine cork no longer fits. We really do not prescribe it for the flu anymore.
But for board exam, the student must remember M2 is the target. It is an ion channel, and it is A only.
Absolutely. High yield fact.
Okay, we have built the machine piece by piece. Now let's watch it run. Segment 2, the replication cycle. Because this is where influenza just completely breaks all the classical rules of virology.
It really does ignore the textbooks. So step 1 is attachment. The HA grappling hook binds to the sialic acid. But here is a molecular detail that separates the novices from the virology experts. The linkage.
Oh, this is the chemistry part. Murray goes into alpha-2,3 versus alpha-2,6 linkages. Why on earth does a medical student need to care about the sugar chemistry?
Because this tiny chemical bond is the exact molecular key to zoonosis. It is the only reason bird flu stays in birds and human flu stays in humans most of the time.
Okay, break that down for me.
Sialic acid is a sugar molecule. It connects to another sugar called galactose on the cell surface. In humans, that connection between the two sugars is shaped like an umbrella. It points up and outward. That is the alpha-2,6 linkage.
An umbrella.
Your entire upper respiratory tract, your nose and throat, is covered in a forest of these umbrellas. And the human flu HA grappling hook has evolved over thousands of years to perfectly grab an umbrella handle.
Okay, and what about birds?
Birds have an alpha-2,3 linkage. Biochemically, that bond is shaped more like a narrow cone.
So the avian flu HA grappling hook is specifically designed to only grab the cone.
Exactly.
So if I walk through a park and breathe in a bunch of aerosolized bird flu from a pigeon, the bird virus goes into my nose, its grappling hook looks for a cone, but all it hits are umbrellas.
As it bounces right off, it cannot attach. You do not get sick.
But humans do occasionally catch bird flu. We see the terrifying headlines about H5N1. How does that happen if we only have umbrellas?
Well, here is the terrifying caveat. Humans actually do possess some of those avian cone receptors, but they are located extremely deep down inside the lungs, way down in the delicate alveoli where gas exchange happens.
Oh, wow. So to actually catch bird flu, I have to inhale a massive viral load deeply enough that it bypasses all the umbrellas in my throat and reaches the cones in the deep lung tissue.
Exactly. That is why H5N1 bird flu is so incredibly hard to catch from casual contact.
But if you do catch it,
the virus sets up shop directly in the deepest, most critical part of your respiratory system. It destroys the alveoli directly, which is why the mortality rate for H5N1 is so staggeringly high. It causes profound primary viral pneumonia.
that makes so much sense. Structure dictates strategy again. Okay, so the virus binds successfully. It gets swallowed into the endosome. The M2 airlock acidifies the core, and the 8 RNA cards are released into the cytoplasm.
Right.
Now comes the rule breaking, because most RNA viruses just stay right there in the cytoplasm. The ribosomes are there, the amino acids are there. Why go anywhere else?
Most RNA viruses think the cytoplasm is the kitchen, so they just start cooking. But influenza completely ignores the kitchen and goes straight into the master bedroom. It travels all the way into the host cell nucleus to replicate.
Into the nucleus. Why? What does the nucleus have that the cytoplasm does not?
It has the host cell's freshly made mRNA. And specifically, it has the caps.
This brings us to the concept of cap snatching, which honestly sounds like a petty street crime.
It is more like high-level molecular identity theft.
How does it work?
Every single piece of messenger RNA your host cells make has a special methylated 5' cap attached to the front of it.
A 5' cap.
Think of it like a VIP security badge. When your ribosomes see that cap, they say, this is an official verified cellular message. I will translate this into a protein.
But the influenza RNA does not have that VIP badge.
No, it does not, so it steals yours. The viral polymerase, specifically the PB2 subunit, physically grabs onto a piece of your host mRNA in the nucleus. Then the PA subunit of the polymerase acts like a pair of molecular scissors.
It just cuts the cap right off.
It snatches the cap off your message and chemically glues it onto the front of its own viral RNA segment.
So it decapitates the host's instructions and wears their hat as a disguise to trick the ribosomes.
It wears the VIP badge. The ribosomes see the cap, and they happily start manufacturing viral proteins instead of host proteins.
That is incredibly sinister. And it accomplishes two things at once, right? Because not only does the virus get its proteins made, but what happens to the host mRNA that just got its head chopped off?
It gets degraded. It is destroyed. Which means the host cell suddenly cannot produce its own defensive proteins anymore. It cannot make interferon to warn the neighboring cells.
It silences the cellular alarm system while stealing all the resources.
Diabolically efficient. And the reason you need to know this is because we now have a very modern drug that targets this exact step.
Baloxavir marboxyl.
Yes, baloxavir. It specifically inhibits that cap-dependent endonuclease activity. It literally breaks the molecular scissors.
So the virus cannot steal the VIP badge and it cannot get translated.
Exactly. It is a brilliant example of how truly understanding the deep molecular mechanics leads directly to life-saving pharmacology.
Okay, so replication is happening in the nucleus. The 8 segments are copied. They get sliced up using the host's nuclear splicing machinery, which is another reason it needs the nucleus. And then everything ships back out to the cytoplasm. The new virus particles assemble at the cell membrane, and they prepare to bud out.
And pay attention to the specific location of that budding. They bud out of the apical surface of the epithelial cells.
The apical surface, meaning the side facing the airway lumen, the empty space.
Right.
Why does that distinction matter clinically?
If the virus were to bud out of the basal side, the bottom of the cell, it would go directly into the underlying bloodstream.
And that would cause viremia, virus in the blood.
Which would be a systemic blood infection.
That's
But by budding apically out into the open airway, it stays entirely within the respiratory tract. It prioritizes being coughed out so it can infect a brand new host.
It wants to spread horizontally to other people, rather than spreading vertically deep into your blood.
Exactly. Which explains perfectly why the flu is almost always a localized respiratory illness and not a full-body blood infection.
Okay, we have the machine built and we know how it copies itself. Now let's move into segment 3, genetics and epidemiology. How this machine evolves over time. We touched on the deck of cards metaphor earlier, but we need to formalize this for the boards. Drift versus shift.
This is probably the highest yield concept in the entire chapter. Let's start with antigenic drift. I call this the annual nuisance.
This is the reason I have to go to the pharmacy and get a new flu shot every single October.
Correct. Remember that viral RNA polymerase we talked about, the photocopier?
Yeah, the P8, PB1, PB2 complex.
Right. Well, it's a very sloppy machine. It does not have a backspace key. It has absolutely zero proofreading ability.
So as it is rapidly copying those thousands and thousands of RNA letters, it is just constantly making random mistakes.
Lots of mistakes. Point mutations, a typo here, a typo there.
And over the course of a year, spreading through millions of people, those typos accumulate.
Specifically, they accumulate in the genes coding for the HA and NA proteins on the surface, so the physical shape of the grappling hook changes just a tiny, tiny bit.
Maybe the handle of the umbrella grabber is a slightly different shade of red this year.
Exactly. So your antibodies that you made from last year's flu shot still recognize it mostly, but not perfectly. They kind of loosely bind, but they do not neutralize it completely.
So I still catch the virus, I still get sick for a few days, but I do not end up on a ventilator because my immune system had a partial head start.
Right. Drift causes seasonal epidemics, just the normal winter flu season. And importantly, antigenic drift happens in both influenza A and influenza B. They both have sloppy polymerases.
Okay, so drift is just a typo in the manuscript. It happens every year.
Yeah.
Now, compare that to antigenic shift, the global killer.
Shift is the process of genetic reassortment. We are no longer just changing a single letter or a typo. We are violently swapping out entire chapters of the book at once.
The deck of cards being shuffled.
Yes. But as we established earlier, to shuffle 2 decks together, you absolutely must have a dual co-infection in the exact same cell.
Enter the pig.
The pig, the ultimate biological mixing vessel.
Why the pig, though? What makes swine so special in the virology world?
Think back to our receptor conversation. Humans mostly have the umbrella receptors alpha-2,6. Birds mostly have the cone receptors alpha-2,3.
Let me guess. Pigs have both.
Pigs are absolutely covered in both. The epithelial cells in a pig's trachea possess huge amounts of both the human umbrellas and the avian cones side by side.
So a single pig on a farm can catch a mild bird flu from a duck pond via a cone receptor and catch a human seasonal flu from the farmer via an umbrella receptor on the exact same afternoon.
Exactly. Both viruses travel down into the pig's lung. They both enter the exact same epithelial cell. They both uncoat and travel to the nucleus.
Now you have 16 loose RNA segments floating around in the soup.
And when the cell starts mass-producing new viruses, they just grab any 8 segments.
So you could randomly get a virus particle that packages the internal engine proteins of the human flu. So it replicates incredibly well in mammals.
Right.
But it accidentally packages the segment for the avian HA grappling hook.
A surface protein that no human immune system on the planet has ever seen before.
A wolf in sheep's clothing.
Or a bird killer in a highly transmissible human package. Because nobody has any pre-existing antibodies to that avian HHA, it sweeps through the entire human population like wildfire. Nobody is immune. That is a pandemic.
And because this requires an animal reservoir to provide those novel gene segments, this only happens in influenza A.
Shift is exclusively influenza A. Because influenza B does not infect birds or pigs in any meaningful way. There is no mixing vessel out in nature for B.
Let's apply this concept to a real historical event, the 2009 H1N1 swine flu pandemic. Murray's text describes that specific strain as a triple reassortment. What does that mean?
It means it was a genetic Frankenstein's monster. When virologists sequenced it, they found it contained gene segments from North American swine, Eurasian swine, avian sources, and human sources, all combined into one virus.
So it had been cooking out there in the pig populations for years, just quietly swapping cards and refining its deck over multiple infections until it finally hit the absolute perfect combination to jump back into humans and spread globally.
Exactly.
And one of the most terrifying things about that 2009 pandemic was the demographic it hit. Paradoxically, it was putting young, healthy adults in the hospital, but sparing the elderly. Usually the flu kills the very old and the very young. Why was 2009 backward?
That phenomena is a concept called original antigenic sin, or immune imprinting.
Original antigenic sin.
It turns out an older H1N1 strain that was somewhat structurally similar to the 2009 swine flu had circulated decades earlier, back in the 1940s and 50s.
Oh, I see where this is going.
Yeah, the elderly population in 2009 still possessed deep memory B cells from their childhood infections that recognized that specific H1 structure, so they had a hidden layer of protection.
But young people born after, say, 1957 had only ever been exposed to H3N2 or H2N2 over the years. They'd never seen an H1 quite like this.
So they were immunologically naked against it. Their immune systems completely overreacted, causing massive inflammation.
Which perfectly transitions us into segment 4.
Yeah.
Pathogenesis, the actual war happening in the lungs. When the virus is replicating, what is it actually doing to the tissue to cause all these symptoms?
The primary battlefield is the ciliated epithelial cell. The virus enters the cell, replicates wildly, and eventually triggers cell death. The dead cell then physically sloughs off the airway wall, a process called desquamation.
I always like to use the analogy of mowing the lawn. Your airways are lined with these microscopic hair-like cilia that are constantly beating upwards, sweeping mucus and dust and bacteria out of your lungs.
The mucociliary escalator.
Right. Influenza comes in like a lawnmower and just mows that beautiful ciliated lawn straight down to the bare dirt.
That is a phenomenal visualization because that is exactly what it looks like under a microscope. You completely lose your primary physical defense mechanism.
The mucus just sits there pooling in your lungs.
Which is why you develop that dry, hacking, severe cough.
Yeah.
Your body is desperately trying to clear the mucus, but the escalator is broken. But the real danger is the vulnerability it creates.
You mean the secondary invasion?
Exactly, or secondary bacterial pneumonia. This right here is what actually kills the vast majority of people during a flu epidemic.
Not the virus itself.
Usually no. A classic clinical vignette is a patient gets the flu, they have fever and aches for 4 or 5 days. Then they actually start to feel a little bit better.
The biphasic course?
Right. The fever breaks, they think they are recovering. But then 2 days later, they suddenly crash. Massive high fever, shaking chills, and now they are coughing up thick, productive green or bloody sputum.
Because the bacteria were just waiting at the gates for the lawnmower to finish its job.
The crazy part is the bacteria were already inside you. They live harmlessly in your nose and throat all the time. Streptococcus, pneumonia, Haemophilus, influenza, Staphylococcus aureus.
But normally they cannot get deep into the lungs because the cilia keeps sweeping them away.
But now the cilia are gone. The raw damaged tissue is exposed. The bacteria just casually walk down the stairs and colonize the deep lung tissue, causing massive lobar consolidation.
Jarrett's really emphasizes one specific bacterial partnership here, the Staphylococcus aureus synergy. And I know this is a huge favorite for board examiners.
It is incredibly elegant and absolutely lethal. So we established earlier that the viral haemagglutinin in the HA has to be cleaved to become fully active, right?
Yeah, it gets cleaved so the grappling hook can unfold and fuse with the host membrane. And usually host cellular enzymes do that cleaving.
Well, Staphylococcus aureus is a bacteria that naturally secretes its own very powerful proteases into the environment.
Wait. So the bacteria is secreting an enzyme tool that accidentally perfectly activates the virus.
It is not an accident. It is synergy. The staph protease cleaves the flu HA. This hyperactivates the influenza, allowing the virus to replicate much faster and destroy more tissue.
And the more tissue the virus destroys, the more dead cellular debris is created, which is exactly what Staph aureus wants to feed on and grow. So they are literally helping each other. I scratch your back, you kill my host.
It is a synergistic death spiral. If you have a patient recovering from the flu who suddenly deteriorates with a rapidly cavitating pneumonia on their chest x-ray, you must immediately suspect Staphylococcus aureus, potentially MRSA.
That is such a vital clinical pearl. Let's pivot slightly to segment 5. Clinical syndromes and complications. We know what it looks like in an adult. The fever, the severe aches, the dry cough. How does this presentation differ in pediatric patients?
Kids are clinically distinct. First off, they tend to run much higher fevers, sometimes high enough to trigger febrile seizures, which is terrifying for parents.
But symptomatically, there's a big difference in the gut, right?
Yes. Kids frequently present with prominent gastrointestinal symptoms, severe vomiting, and abdominal pain.
Which you almost never see in a standard adult flu case.
Yeah. Right. If a 30-year-old comes to the clinic saying they have the stomach flu, it is almost certainly a norovirus or a foodborne illness. It is rarely influenza. But if a 5-year-old is vomiting, influenza is absolutely on the differential.
What other pediatric complications are common?
Because their airways are so small, they frequently develop croup. That is acute laryngotracheobronchitis.
The classic barking seal-like cough.
And a lot of otitis media, too.
Yep.
Ear infections, because the swelling blocks the eustachian tubes.
Now, we have to issue a very serious clinical warning here. Something every medical student has drilled into their head. Rey syndrome. We put it in the outline, but I really want to understand the deep mechanism behind this. Why is it an absolute cardinal rule that we never give aspirin to a child with the flu?
Rey syndrome is a catastrophic metabolic disaster. It specifically occurs when you administer salicylates like aspirin to a child who is currently fighting off a viral infection, primarily influenza or varicella chickenpox.
Why those viruses specifically?
We actually still do not fully understand the exact viral trigger, but we know exactly what the aspirin does. The salicylates physically damage the mitochondria in the child's cells. They act as uncouplers of oxidative phosphorylation.
The powerhouse of the cell is completely shut down.
Exactly. Without functioning mitochondria, the liver cannot process waste. You get a massive accumulation of ammonia and free fatty acids in the blood.
And where does all that toxic ammonia go?
Straight to the brain. The clinical result is twofold. First, you get microvesicular fatty liver. The liver fills with fat droplets and fails. And second, you get acute severe encephalopathy.
Brain swelling.
The child becomes extremely lethargic, confused, they start vomiting violently, and they quickly slip into a coma.
And the mortality rate for Rey syndrome?
It is devastatingly high, up to 40%. And even those who do survive are often left with permanent severe neurological damage.
All from an over-the-counter pain pill.
Which is why the rule is absolute. You never, ever give aspirin to a febrile child. You tell the parents to use acetaminophen Tylenol or ibuprofen Motrin. Never aspirin.
Crystal clear. Let's briefly touch on one more severe complication we mentioned earlier. The H5N1 bird flu clinical case from Murray. You said it has a 50% mortality rate. But if it is so hard to catch because it only binds to those deep lung cones, why is it so uniformly deadly when it does take hold?
Because when it finally does get down into those deep alveolar sacs, it triggers a massive, uncontrolled cytokine storm.
A cytokine storm. The immune system basically panics.
It completely panics. The virus is destroying the delicate gas exchange tissue, so the immune system floods the lungs with inflammatory cytokines like interferon alpha and interleukin-6.
And what does that do physically to the lung?
It makes all the blood vessels in the lung extremely leaky. Blood plasma and inflammatory cells rush into the alveoli, filling them with fluid.
Acute respiratory distress syndrome.
BRDS.
ARDS.
Yes. You essentially drown in your own extreme immune response. So standard seasonal flu typically kills you indirectly via secondary bacteria or by exacerbating an underlying heart condition. But bird flu kills you directly by explosive primary viral destruction and immune overreaction.
It is a totally different mechanism of death.
Precisely.
Okay, let's move to the solutions. Segment 6, diagnosis, treatment, and prevention. How are we actually finding this virus in the clinic?
Well, during the peak of winter, you can usually just guess based on the clinical symptoms. But for official diagnosis and for the boards, the gold standard is RT-PCR.
Reverse transcription polymerase chain reaction.
Yeah.
Because it is an RNA virus.
Right. PCR is incredibly sensitive, incredibly specific, and it easily differentiates between influenza A and influenza B.
What about the rapid antigen tests? The ones you get at the urgent care, they give a result in 15 minutes.
They are very fast and very convenient, but you have to be careful because they have lower sensitivity.
Meaning false negatives are common.
Very common. If a patient feels like they got hit by a truck and it is January and the rapid test is negative, you still treat them for flu. You trust the clinical picture over a negative rapid test.
Now, Murray describes an old-school virology technique called haemadsorption. How does that work?
It is actually a really beautiful visual technique. If you take a sample from a patient and grow it in a cell culture in a petri dish, the infected cells will start manufacturing those viral haemagglutinin spikes and expressing them on their own surface.
Before the virus even buds out.
Right. Then you literally just pour a liquid suspension of guinea pig red blood cells over the culture plate and wash it off.
And what happens?
Because of the HA grappling hooks, the red blood cells stick tightly to the surface of the infected cells. If you look under a microscope, it literally looks like red sprinkles stuck to a donut.
They're sprinkles on a donut.
If you see the sprinkles, you know definitively that you have a hemagglutinating virus growing in that culture.
Yeah.
Like influenza, parainfluenza, or mumps.
I will definitely never look at a donut the same way again. All right, let's talk pharmacology. How do we fight back? We already mentioned the machete blockers.
Right. The neuraminidase inhibitors, oseltamivir, which is the oral pill, and the namamivir, which is an inhaled powder.
And their mechanism is just jamming the machete.
They are structurally analogs of sialic acid. So the viral neuraminidase thinks it is cutting sialic acid, but instead the drug permanently wedges itself into the active site of the enzyme and jams it.
So the newly replicated virus particles just clump up on the dead cell and cannot spread.
Exactly. These drugs work very well against both influenza A and B, but timing is absolutely critical.
You have to give it early, right?
You must administer it within the first 48 hours of symptom onset.
Why 48 hours? Why not on day 4?
Because by day 4, the virus has already reached peak replication. It has already infected most of the cells it is going to infect. The damage is done. Blocking release at that point does not help much. You have to give it while the virus is still actively spreading from cell to cell.
Got it. What about the cap-snatching blocker we talked about?
Baloxavir marboxyl. It is newer. It directly inhibits the polymerase acidic PA protein. It stops the endonuclease activity so the virus cannot steal the host mRNA cap.
And the benefit of baloxavir?
It is a single-dose pill, which is fantastic for patient compliance compared to 5 days of Tamiflu. And it also works against both A and B.
And finally, the old escape hatch blockers, amantadine and rimantadine.
They block the M2 ion channel, preventing the virus from uncoding in the endosome.
But again, these are functionally obsolete for flu, right?
Yes. They only ever worked on influenza A because B lacks the M2 protein. And now almost all strains of A are highly resistant. We really do not use them for antivirals anymore.
Although amantadine has a totally different second life in neurology, doesn't it?
It does. It actually crosses the blood-brain barrier very easily, which caused a lot of central nervous system side effects in flea patients like dizziness and ataxia.
But they found out it helps with Parkinson's disease.
Exactly. It stimulates the release of dopamine in the brain, so it is still a very heavily tested drug on the boards, just usually in a neurology context instead of microbiology.
Okay, last up in our defense arsenal. Prevention.
Yeah.
The annual guessing game. The vaccine.
We basically have 2 main flavors of the flu vaccine. The first is the inactivated killed vaccine. This is the standard shot in the arm.
And what is actually in the syringe?
It is usually a mixture of just the highly purified HA and NA proteins from the virus, a subunit vaccine. Sometimes it is a split virus where they just break the whole virion apart with detergent.
But it was totally dead.
Totally dead. It cannot possibly give you the flu. It generates a strong serum IgG neutralizing antibody response in your blood. It is very safe for everyone, including pregnant women and immunocompromised patients.
But you still ask people if they are allergic to eggs before you give it.
Right, because historically the virus is grown in millions of chicken eggs to mass produce the vaccine. There are cell-based options now for people with severe A allergies, but it is still a standard screening question.
Okay, so that is the shot. What about the live attenuated vaccine? The nasal spray. Flumist.
This one is a marvel of genetic engineering. It is a live virus, but it is a temperature-sensitive mutant.
Temperature sensitive. Meaning what?
The scientists mutated the viral polymerase so that it works perfectly fine at 25 degrees Celsius, but it completely denatures and dies at 37 degrees Celsius.
Wait, 25 degrees is room temperature. That is the temperature of the air inside my cool nasal passages.
Exactly.
But 37 degrees is my internal core body temperature. That is the temperature of my deep lungs.
So when you spray this live virus into your nose, it replicates beautifully in the cool nasal mucosa. It triggers a massive local immune response, specifically mucosal IgA antibodies, right at the front door.
But if the virus tries to travel down your trachea into your warm lungs to cause pneumonia,
It physically cannot survive the heat. The polymerase melts, it stops replicating, and it dies harmlessly. It is brilliantly cold adapted.
that is genius. But because it is a live replicating virus, there are contraindications.
Yes, you absolutely cannot give the live nasal spray to anyone who is immunocompromised, to pregnant women, or to the very young or very old. Their immune systems might not handle even the mild nasal replication well.
All right, we have covered an absolutely immense amount of ground today. From the loose-leaf segmented genome to the sialic acid handshake to the mitochondrial toxicity of aspirin. Now it is time to see if you, the listener, were actually paying attention.
It is time for the Learning Reinforcement, the SBA quiz. I'm going to pose a quick clinical scenario or a mechanistic question. I want you to pause the deep dive, think of the answer, and then talk me through it.
Let's do it. I'm ready. Hit me.
Question 1. A 35-year-old male presents to the clinic with severe flu-like symptoms. A rapid test confirms he has influenza B. A first-year medical student suggests prescribing amantadine to shorten the duration of the illness. Why is this drug absolutely contraindicated mechanistically in this specific patient?
Okay, amantadine. That is the cork, the escape hatch blocker. It blocks the M2 ion channel to stop uncoding, but the test confirmed influenza B, and we learned that influenza B simply does not have an M2 channel. It uses a totally different mechanism. So the drug literally has 0 targets to bind to in this patient.
Spot on. Influenza B lacks the M2 protein. Amantadine is entirely useless here.
Next one.
Question 2. We talked extensively about the difference between seasonal epidemics and global pandemics. Which specific structural or genetic feature of the Orthomyxoviruses family is the absolute prerequisite for the generation of a pandemic via antigenic shift?
It has to be the segmented genome. If the RNA was just one long single scroll, you could not easily swap out huge chunks of genetic information. But because the genome is a deck of 8 separate loose cards, you can have that massive reassortment event when a pig gets co-infected.
Correct. The segmented genome allows for genetic reassortment, which directly drives antigenic shift.
Hit me with another one.
Question 3. A 7-year-old boy seems to be recovering from a bout of the seasonal flu, but his mother gives him an over-the-counter medication to help with the lingering fever. 12 hours later, he develops severe vomiting, acute confusion, and slips into a coma. Lab tests show highly elevated liver enzymes and serum ammonia. What is the diagnosis and what was the medication?
Oh, this is the absolute nightmare scenario every pediatrician dreads. The diagnosis is Rey syndrome. The mother gave him aspirin or salicylate. And the drug caused severe mitochondrial toxicity, leading to acute microvesicular fatty liver and fetal cerebral edema.
Perfect recall. Rey syndrome from salicylates.
Give me a tricky mechanism one.
Question 4. Let's look inside the host cell. Where specifically in the host cell does the influenza virus replicate its RNA genome? And why is this location considered highly unusual for this type of virus?
Influenza breaks the virology rules. It replicates entirely inside the nucleus of the host cell. This is highly unusual because almost all other RNA viruses just replicate out in the cytoplasm. And the reason it goes to the nucleus is to perform cap snatching. It steals the 5' cap from the host's newly synthesized mRNA to use as a primer for its own viral translation.
Excellent. Cap snatching in the nucleus.
Let's do the synergy one.
Question 5. An elderly patient with a confirmed case of influenza seems to be recovering well. However, 1 week later, he returns to the emergency department with a very high fever-productive bloody sputum and severe lobar consolidation on his chest x-ray. A sputum culture rapidly grows Staphylococcus aureus. How did the bacteria mechanistically assist the virus during this co-infection?
Okay, this is the synergistic death spiral. The Staphylococcus aureus secretes a powerful protease enzyme into the lung environment. That specific protease effectively cleaves the viral haemagglutinin HA on the newly budding flu viruses. Cleaving the HA hyperactivates the virus, allowing it to fuse and infect many more neighboring cells much faster than it could on its own.
Outstanding. That is a really high-level board distinction.
Last question. Bring it home.
Question 6. What is the exact biochemical function of the neuraminidase envelope protein, and why is this function absolutely necessary for viral spread?
Neuraminidase is the machete. Its biochemical function is a sialidase. It cleaves sialic acid residues off the surface of the host cell. This is necessary to prevent the newly formed budding viruses from getting stuck to the dying host cell or from clumping together in a sticky mass. It chops the tether, allowing them to release and spread.
You nailed it, 100%.
Well, there you have it. The Orthomyxovirus, a masterclass in evolutionary adaptation and molecular warfare.
Before we wrap up, I want to leave you with one final provocative thought to chew on. We spend so much of our time on our global surveillance resources worrying about the pig.
Right. We worry about the mixing vessel, the antigenic shift.
We constantly monitor for a massive reassortment event like H5N1 swapping cards, but I want you to look back at the 1918 Spanish flu pandemic.
The biggest viral killer in modern human history. It killed, what, 50 million people?
At least. When virologists finally dug up victims from the Alaskan permafrost a few decades ago and actually sequenced that 1918 virus, they found something deeply disturbing.
What did they find?
It was not a reassortant. It did not look like a messy mix of pig and human and bird cards. It looked like a pure, untouched avian virus that had just slowly adapted. Mutation by tiny mutation.
Drift.
Pure antigenic drift. It just drifted until it perfectly unlocked the human respiratory tract.
So you do not always need a massive, sudden shift to create a global monster. Sometimes a slow, steady drift can stumble upon a combination that is just as deadly.
Exactly. The virus is always learning. It does not always need a shortcut. Sometimes it just puts in the slow evolutionary work.
Wow. Well, that is a genuinely terrifying thought to end on, but a very necessary reminder of why we study this. If you want to visually see exactly how that nuclear cap snatching works, that molecular identity theft, I highly encourage you to open up your copy of Murray Chapter 49 and look closely at Figure 49.2. It is a fantastic diagram that maps the whole cycle out beautifully.
And as always, a quick reminder that this deep dive is strictly for educational purposes. We are not your treating physicians. Always consult the primary textbook for your actual medical exams.
Thanks for listening to this deep dive. Wash your hands, get your seasonal flu shot. Please don't give aspirin to kids, and we will see you next time.